So while we make the transition, I will introduce our next speaker, Professor Kramer. So Professor Kramer, let me just orient myself here. Professor Kramer obtained her honors BS in chemistry from the University of Utah and her PhD in organic chemistry from UCLA under the direction of Tim Deming. She then completed an NIH NRSA postdoctoral studies at Stanford and UC Berkeley as a UC Chancellor's postdoctoral fellow in the lab of Professor Carolyn Bertozzi. She has also spent several years in industry with Echelon Biosciences. Dr. Kramer began her independent career at the University of Utah in the Department of Biomedical Engineering in 2017. She's the recipient of the Dream Chemistry Award, an NSF Career Award, and an NSF Rapid. Her lab utilizes a combination of polymer chemistry, protein engineering, and bioconjugation techniques to design new bio-inspired materials and to probe fundamental questions in glycobiology while addressing human health needs. So it's my pleasure to introduce to you Dr. Kramer, and the title of her talk is Synthetic Mucins from New Chemical Routes to Engineered Cells.
So I'm very excited to tell you today about some of the new projects that my lab has been working on. We are, of course, a newer lab since I just started my position in 2017, but we've made some really nice headway on our goal of synthetic mucins and understanding mucus biology. So I'll start out with some background on mucins and why we got interested in studying the mucin Glycocalyx and mucus as a material. So mucus is this coating that we're all familiar with, as the secreted substance. The major protein component of mucus is a glycoprotein called mucins. And so these are secreted and they can be a few hundred microns thick on the tissue surface or expelled, as we're familiar, but also these mucin proteins are tethered to the cell surface to compose the mucus Glycocalyx. And this is an incredibly huge surface area of the human body. We actually have about 200-fold more surface area of mucus and mucin coated epithelia than skin. And this is found in the stomach, eyes, lungs, reproductive tract, and even on the heart, which is the picture that I'm showing down there in this part of the screen. You can actually see the mucin Glycocalyx in this SEM. And so there's 20 plus known mucin genes. So we have a bunch of different protein sequences for mucins. And of course, some of these are tethered to the cell surface and transmembrane to become the Glycocalyx, and some are secreted. They serve incredibly important functions in lubrication, hydration, filtration. So all of the gases and nutrients and things that we absorb are filtered through the mucus. And as such, they are sort of interfacing with the outside world. And this mucus and mucin substance is a reservoir of immune cells in the microbiome. So incredibly important, and that's sort of why I got interested in mucin structures. And as you can imagine, when mucins are misregulated, we see disease states, such as cystic fibrosis with a very thick and sticky mucus, premature birth, where the mucus plug at the cervical opening is, if it's compromised, there can be infection and premature birth, dry eyes, gastric infections. And what had first sparked my interest in understanding mucin biology was actually epithelial cancers, when I had learned that 90% of epithelial cancers have abnormal mucin structures. And so this is both glycosylation patterns and expression levels. This was really interesting to me because these are really disparate tissues, like lung tissue, colon tissue, breast tissue. These are very different types of tissues in the body. But they have the same phenomenon of abnormal mucins. So I got interested in understanding why these structures are different and what biological effects that has. So what are the structures of mucins? Showing the cartoon version with a little zoom in to the chemical structure. And so mucins are a protein backbone. And there are a variety of core glycan structures attached to serine and threonine residues. And so for most of my talk, I'll use our glycan, what in the Bertozzi lab we called the lucky charms of the glycan structures. So you'll see those cartoons throughout the rest of my talk. But mucins have these unique glycan patterns, and they will vary between different species. So mucus is a very, it's almost like a primordial lubricant substance. So even snails and jellyfish have mucus, and we see different glycosylation patterns and different peptide sequences in these different species, but also even in between individual humans. So you and I may not have the same mucin structures, and this will vary with disease states, as I just mentioned. And so overall, the glycosylation is quite heterogeneous. There are over 1,000 different enzymes that are involved in both the metabolic processing of dietary sugars and conversion of one sugar to another, and then attaching these sugars to the peptide backbone. So this is very complicated, and we as glycobiologists do not have a full picture of the pathways of how these mucins get formed. And so that's made studying them actually quite difficult. We can't obtain homogeneous samples where we have a mucin with a certain set of glycan patterns that we can then study the biological outcomes. So we can't obtain those from biological sources. And so this is sort of where my chemistry background came in, and I was looking at the state of studying mucins, and actually most people are using farm animal mucins, where intestines or tissues or submaxillary glands are scraped, and mucin samples are obtained from those scrapings. And so they're crudely purified and separated from some other proteins, but they become denatured. And of course, the materials you get are heterogeneous in their glycan patterns. And so, people have complained that if you buy mucin from Sigma, you are getting this whole mixture of compounds. And if you buy the bottle again 6 months later, your experiments may not give the same outcome. And of course, if they come from animal sources, they won't have human glycosylation patterns. And there's similar challenges to recombinantly producing mucins. It depends on the cell type and the age, passage number of the cells. And so you can get different outcomes and glycosylation patterns there. And then the other method that people will use to try to get mucins is peptide synthesizers. And of course, you can obtain homogeneous, very pure samples this way, but they are just fragments. They're very small. Peptide synthesizers are, this process is not very efficient, especially with glycosylated amino acids. So you tend to get things that are, say, 10 to 20 residues long, whereas natural mucins are hundreds and even thousands of residues long. And so this is where my background came in, where I had been working in polypeptide synthesis research. And we thought our synthetic polymers or our polypeptides could actually solve some of these challenges for mucin research. The method that my lab uses is called NCA polymerization for N-carboxy anhydrides. So we take amino acids and we treat them with phosgene or some sort of phosgene equivalent, a halogenating reagent, and cyclize them to these 5-membered rings or NCAs. And there's a variety of ways to do ring opening polymerization where you end up losing CO2 and gaining a polypeptide. And this can be done with nucleophiles. We use transition metal initiators because they are very rapid in their kinetics. And so we can get polypeptides that are hundreds of residues long in about an hour. And this is also scalable. We can do this on the milligram to multi-gram scale. And this is actually used industrially to produce a multiple sclerosis medicine. So this is industrially scalable as well. The nice part about the transition metals is that we can get very long chain lengths with very low dispersity. So meaning that the chain lengths that we get are all very uniform. We don't have a mixture of short and small polymers. And overall, the polypeptide backbone harnesses the physical properties of proteins. So you can get structures like alpha helices and beta sheets. and things that you would get from natural proteins, but we have these advantages of synthetic polymers where we can produce them very rapidly and on scale and with, for example, uniform glycosylation patterns, which is what I'd like to show you on this slide. I've actually been at this technique for quite a while to produce glycopolypeptides. It's something that I had started working on even as a graduate student, where I got interested in using this polymerization technique with glycosylated monomers. And so on this slide, I'm showing a variety of different glycan structures that we've utilized with this chemistry. Some of these are natural linkages, such as manobiose, manoserine, galnacserine, lacserine. Some of these are not natural linkages, such as with cysteine or with lysine. And some of these we had designed specifically to be more stable to enzymatic deglycosylation or more stable to hydrolysis in acidic conditions. And because we are making these synthetically, we really can tune the reactivity and we can feed in monomers bearing different sugars and end up with a glycopolypeptide displaying these, say, natural or unnatural sugars in varied ratio. And we can tune the density and the pattern. And so that's just not something that can be achieved using biological methods to obtain mucins. So I wanted to show just a few pieces of data that highlight the power of this technique for us. This structure here is alpha galnacserine monomer, and this is the core structure found in mucins. This is the initial glycan that is found on all mucins. And we had designed this nickel initiator shown here because it bears an azide group. And as our previous speaker so nicely showed us the power of azides for click chemistry, we wanted this so that we could have a synthetic mucin with an azide on one end and the amine terminal on the other end. And therefore we could attach say fluorophores and a cell anchor or a target of some sort for our biological work. So that was the purpose of that one. The data down here at the bottom of the slide is showing some of the properties of these synthetic mucin polymers. And this is plotting the monomer to initiator or NCA to nickel ratio and the molecular weight of the polypeptide we obtain. And you can see this nice linear trend. And so we can predetermine or tune the length of the synthetic mucin that we get simply by varying the NCA to the nickel ratio that we feed into the reaction. And we can obtain structures of this DP is degree of polymerization or chain length. And so we could get nearly 400 amino acids in this chain where every single one has a galnacserine, if that's what we choose. And actually in natural mucins, it depends on the mucin gene. Some of these structures are like 90% proline, which gives rigidity to the mucin structure. But there will be 90% proline and glycosylated threonine and serine in some of these structures. It's quite incredible, the density of glycosylated amino acids in mucins. So we can really tune that from 100% down to whatever we like simply by blending in amino acids that don't have sugars on them. Now, one of the remaining challenges, sorry, I got ahead of myself, is glyco brushes. And so I had showed you that picture with the mucin where we have up to 15 different sugars or 15 units in the sugar side chain. So they make these bottle brush-like structures. And in the previous slide where I showed you all those different sugars, we had done this chemistry with a trisaccharide, actually. But beyond that, it's actually rather difficult to purify the monomers and they're very sterically demanding. And so I don't think we can make a monomer with a 15-mer sugar and try to polymerize it. There is a limitation there. So we wanted to try to make these brush-like structures and started thinking about techniques in brush polymer chemistry. And the main ways, this is a review from Summerlin and Matuszewski. The main way that people are making brush structures are mostly grafting to method, where you take a functional polymer. So you already have an existing polymer and it has some kind of activatable group on the side chain or some click group, and you click this to a sugar chain. And so this is called grafting 2. And almost all glycobrush efforts have been focused there. The problem is that that's quite inefficient and you really don't get a lot of chains attached. The sterics prevent a high density of side chain attachment. And so people have also looked at grafting from, which is you have an existing polypeptide backbone or a polymer backbone with some sort of activatable group, and you grow, you add a monomer and you grow side chains from it. And in this area, there are a couple examples of people trying to do this to make glycosylated structures, but it's been all with radical polymerizations like ATRP. And so you get a hydrocarbon backbone, hydrocarbon-based side chains. And for us, that's not so appealing because we cannot get the peptide backbone conformation that you would see in a natural mucin. They wouldn't be biodegradable by natural proteases. The orientation and this alpha conformation in the sugar is actually quite important for the structure and also biological recognition. So we needed to develop some new chemistry to attack this problem. And this is the work of a student in my lab, Zach Claus. And so we were inspired by some work from the Deming lab at UCLA and also some of our work on these nickel azide initiators. And what Zach and I did is we knew that we could take these alloc protected amino acids and turn them into these nickel initiated species. And so we knew that alloc amino acids had this reactivity with these nickel complexes. And so what we did is design an NCA monomer that has also this alloc group on it. And so that will be represented by this purple sphere. And so Zach prepared this NCA monomer bearing the alloc group, and we also designed in this methionine linker. And I'll explain to you in just a moment why we chose a MET linker. So Zach polymerized this NCA monomer with a cobalt initiator that does not react with the alloc group. And he formed this polypeptide bearing these activatable alloc groups, which are shown as the purple spheres. And so then we could add a nickel complex. This is dimpy nickel cod, and that will react with the alloc groups to form a complex like this one shown at the top of the slide. And then we can add a second NCA monomer and grow brush structures. And so we did this with a variety of different glycosylated monomers as our secondary monomer to grow the side chains. And so we did this with glucose lysine, a galnacserine, and a lacserine. And we found that these would all polymerize very efficiently. We could actually get side chain lengths from 10 to 300 plus amino acids. Of course, this is bigger than we would find in nature, but these are actually reminiscent of structures like proteoglycans. If people are familiar with proteoglycan structures, they're similar to mucins in that they have a peptide backbone with sugar chains, but the sugar chains are long, up to hundreds of residues. And so we can actually mimic proteoglycan-like structures with this chemistry. Again, the glycan composition and density is tunable based on the monomers we feed into the reaction. We can tune the chain length and how densely packed the bristles of the brush are, if you will. And so the data I'm showing down in the corner here is size exclusion chromatography coupled with 18-angle light scattering refractive index detectors. And so you can see that when we have different backbone lengths, such as a 63 mer versus 75 mer, and we have different side chain, the bristle length 23 versus 50 versus 68 versus 117. These will elute at different times in our size exclusion chromatography. And so we can actually see that they are different molecular weights. And that's one way that we can characterize them. We also use proton NMR, and we've used some other techniques, which I'll show you in just a moment. Okay, now I wanted to point out why we use the MET linker in that structure. And the reason is that peptides can be cleaved at met residues using cyanogen bromide. And that is really key for us to be able to separately analyze the backbone polymer from the polymeric glyco side chains or the bristles of the brush. And we wanted to ensure that we're not growing a peptide backbone and then growing a bunch of short little chains mixed with long chains. And so when we analyze the entire brush structure, we don't get a picture of the dispersity in the side chains. And so that's what we wanted to do here. And so Zach is showing his data where we have the backbone structure, which is the AMK. So that's the alloc-met lysine monomer, compared to the entire brush, which is much higher molecular weight here, and then the cleaved side chains. And so we have the polygalnac serine side chains analyzed separately after cleavage. And for those of you who do any polymer work, you'll notice that this hump over here, this sort of this kind of bimodal distribution that we see, this is actually due to aggregation. Again, we're making mucus-like structures of mucus mimics. We know these proteins are aggregation prone, they are sticky, and so it can be difficult to get them to elute as beautiful, nice peaks and not aggregate and look like they are bigger molecules. Okay, a couple other pieces of data about these structures. This is showing the nice linear growth. So we again have that relationship between monomer and initiator, and showing that by tuning the NCA to nickel ratios, we can grow chains of different lengths. And so this is for the branches, the bristles of the brush after cleavage. We're showing we can grow them at different lengths. And this can happen even if every single unit on the backbone is growing a side chain. So this is like an incredibly sterically demanding molecule. Every single amino acid on that peptide backbone is growing a sugar side chain. And even then we can get linear growth. So it's not affected by whether their branches are spaced or packed. And that's really highlights the fast kinetics and efficiency of this polymerization chemistry. Zach did some AFM studies in collaboration with the Bustamante lab at UC Berkeley, who has a lot of experience in imaging these types of molecules. We could see by dynamic light scattering that if we have a backbone of different lengths, so 150 mer versus a 300 mer, so the backbone itself is twice as long, the bristles are about the same length, and we could see that they're indeed scattering light and giving us different sizes. And Zach also, in collaboration, was able to obtain some direct images and do some statistical analyses on the image to get the particle size. And so he prepared this violin plot showing the size distribution. And we can see again that, as expected, the different chain lengths do give us different particle sizes and morphologies. I wanted to point out some of our biological data and why we are not satisfied with hydrocarbon-based or ATRP-based polymers, and why we bother to do this peptide polymerization chemistry. These are cytocompatible. There are no, even at high concentrations, we did not see any ill effects on the cells. We tested here the galnac serine and the glucose lysine structures. And these brushes did not cause any toxicity. We tried hex cells, and we've also done this in a variety of other cells where I'm not showing the data. So, you know, we think they're quite well tolerated, as expected, since they are mimicking a natural structure and they are based on natural materials. This data is also, again, highlighting we think using a peptide backbone is important. This data is showing biodegradation by mucus-resident microbes. And so many years ago, a lab had published on an esterase, a protease that should be mucin specific. It's found in E. coli. And so this makes sense. E. coli will be in the gut. And in order to form biofilms and colonize, they will chew up the mucus and kind of make their way into the protective mucus layer and even eat some of those byproducts and use them for metabolism. And so it makes sense they have an enzyme that's specific for that. And it was named, not by me, by the lab that published this, sticky, for secreted protease of C1 esterase. And sticky should only cleave the peptide backbone if it's glycosylated with GalNAc. And so that's exactly what we saw. This silver stained gel shows in the lanes 1, 2, 3, 4, this is an enzymatically active sticky up here. And then the E447D is where a catalytic residue has been mutated. So the glutamate is mutated to an aspartic acid. And so this one is catalytically inactive. And then we use trypsin, was just a very general protease. And so you can see here is our just our synthetic mucin. They streak like crazy. These are not ionic, right? They're coated with neutral sugars and they don't interact with SDS very well. And so it's very typical for both our mucin, our synthetic mucins and native mucins to see a big streak up the gel. And so then you can see when we add sticky or trypsin, it's essentially totally degraded. And so they are recognized by natural mucin degrading enzymes. Whereas the catalytically inactive is not degrading the enzyme or degrading our mucin. And then if we take the sugars off with a commercial deglycosylation mix, we see that it's not degraded by anything except for trypsin. So this tells us it's recognized by these natural substances or natural enzymes. Okay, now in the last few minutes, I wanted to tell you about how some of the ways we're using the mucins. And this is a strategy where we're attaching cholesterol to our synthetic mucins. And then we can supplement the media of cells with this lipid anchored synthetic mucin. And we have a dye on there so we can see it. And so you can see that we can sort of coat the cells with this artificial glycocalyx using these lipid anchored polymers. And some of the things we're doing with this glycocalyx engineering strategy are to try to understand how they affect the cancer glycocalyx. As I had mentioned, mucins are abnormal in 90% of epithelial cancers, and this has been noted by the FDA and the NCI, and mucins are a really hot target if we can understand their structures and which ones are most relevant to transformation and cancer progression. And we started working with a lab at the U of U at the Huntsman Cancer Institute. This is Kasia and Sarah Ann from the Rosenblatt lab, and they work together, Kasia's in my lab, to try to understand tumor formation. And so normally when cells are growing and growing, they get crowded, and at some point some of them pop off, and homeostasis is maintained in the layer, and that does not happen with cancer, right? We get a buildup of cells and tumor formation. And so what Kasia and Sarah Ann showed is that if we coat cells with a bulky glycocalyx like that found in epithelial cancers, we can actually slow down extrusion by this whopping threefold reduction. And so what we're showing is essentially just coating the cells with the glycocalyx induces tumor formation. This was really, we were very surprised by these results. Now, in the last minute, I'd like to tell you about our coronavirus project. Of course, the coronavirus must make its way through the mucus and the glycocalyx in order to infect cells. And so early on in the pandemic, I started reading about the interactions of mucus and coronaviruses, and they actually bind to sialic acid structures. And in some coronaviruses, this is this binding event initiates membrane fusion and is the pathway for infection. And SARS-CoV-2 uses a different protein for cellular entry, but it actually does have a mucus binding domain in the spike protein, which we think is very important for docking. And we are really interested in, how people's different mucus composition and their mucosalivary compositions affect viral transfer and viability. And you can really see, I love this picture from the Berubia lab at MIT showing why we all wear masks and the ejection of mucosalivary fluid into the air to generate, contact transfer or fomite objects. And so the subject of the RAPID grant that we have is to understand how these different mucin structures affect transmission and infection, both through airborne droplets onto surfaces and also through the Glycocalyx. And so my fantastic graduate student, Kasha, is working on this project. And so this is her recent picture of culturing human epithelial cells, and these are infected with coronavirus. It's not SARS-CoV-2. We don't have a BSL-3 lab, but we have a BSL-2 plus version of a coronavirus that we can work with safely. And so you can see the white plaques from viral infection in this picture. So with that, I'd like to just point out some of the sort of dreams we have with synthetic mucins. We'd like to create fully synthetic human mucus and study mucosal transport, and hopefully eventually contribute to dry eye treatments or wound healing gels, or potentially even beauty products and artificial snot to enhance sensors. There's a lot of cool things we can do with synthetic mucins. I'd like to thank all the students in my group and all the funding sources. And of course, Utah is a lovely place to be a researcher because we can go skiing or go to national parks. So with that, I'd be happy to answer any questions that you all have.